So I'm really appreciative of the opportunity to be here and speak today and to listen to all the other great scientists and community members. And it's just a real honor. And I come with a bit of a different perspective than I think most scientists and researchers that we're hearing from, and that I was trained in aging and in the neuroscience of how our brains change as we grow older and what factors influence that and how cognition changes. And then was fortunate enough to be presented with this need in the autism community to fill a gap in understanding how aging might uniquely affect autistic individuals. So I paid close attention to this paper that Dr. Piven mentioned yesterday that he led about defining a research agenda. And this was really a guidepost for me as a postdoc in the research program that I wanted to develop. And as I went out into the community, both my local community in Phoenix and surrounding areas, as well as paying attention to conversations happening online, it was so much like what we just heard from our panelists of like, this is something that autistic adults are very concerned about themselves and the ability of our society to care for their unique needs, and parents wondering what's going to happen to my kid when I'm not here anymore. And then as the research has continued to unfold, like we heard from Dr. Shea, we see that indeed this, part of this fear is coming to reality as far as a increased dementia risk associated with autism. So I started my research program about 6-7 years ago, and in my lab at Arizona State University, the Autism and Brain Aging Lab. And I'm going to talk about some of the things that we found over these last few years in kind of two parts. Like, what do we know now from our cohort about the cognitive and brain aging trajectory? And then how can we take what we know about that and try to work towards this goal of precision care, where we're able to figure out the most pertinent things about an individual that's going to predict their future outlook, that's going to be important in how they're going to respond to various interventions. So this study first began funded by the Department of Defense, as well as our local Arizona Alzheimer's Consortium. And in the beginning, for some naivety and some, research control, we started with only middle-aged and older men. And this is something that is not the case anymore. And gender and sex differences is a very big part of my research program now. But back then, we recruited men and neurotypical matches. Everybody's right-handed for some funky things about the way language works in the brain. And in our initial sample, all participants were without comorbid ID, so IQ over the age of 70. And we collect lots and lots of data. We, feel like these participants are just precious, and the ability for them to come spend time with us, we want to take advantage of as much as we can as far as describing as many things about them that might be important to aging. So lots of cognitive assessments of different cognitive domains. We do a little over an hour in the scanner to get different structural scans and functional scans of how the brain is working. And then we ask them a lot of questions about their experiences, both as it relates to traits associated with autism and mental health and quality of life. And when we can, we ask someone who knows them well some similar questions. So the first paper we published on this cohort was just baseline differences, middle-aged and older men with autism compared to neurotypical men who are very similar. And the question here is really, are the cognitive and brain differences that are associated with ASD in children and young adults still present in middle age? When we were starting out, this was still a big question. Do those challenges change? Do people grow out of them? Just what's the picture here? So for cognition, executive function is something that comes up lots with kids, adolescents, young adults with autism. And indeed, in this middle age and older age range, we saw evidence for still persistent executive function challenges. So this is one task called the Wisconsin card sorting task that our autistic adults are making more errors on average than the neurotypical adults. And looking in the brain, a brain structure that I'm going to talk about a lot in this talk is called the hippocampus. And the hippocampus is our primary memory brain structure. And there's kind of mixed evidence about how the hippocampus looks in autistic children and adolescents. But we found pretty compelling evidence that at this middle age point, the hippocampus was smaller in our ASD group compared to our neurotypical group. And again, this is the brain structure we rely on to make memories. We can do another type of brain scan that looks at the way our brain is connected and how different brain regions communicate with one another, at least on the structural level, how those connections look as far as, kind of an ideal biological state. And consistent with smaller hippocampi, we also saw that the connections that were leading out from the hippocampus to other brain regions were showing some impairment in their structural connectivity in the ASD group. So this gave us some important evidence about, yes, cognitive and brain differences that we see in younger autistic individuals are persisting at middle age. And next step is we want to think about aging. How are these things changing across the adult age range? And there's two different ways to do this, which I'll talk about quite a bit, is cross-sectional and longitudinal. So briefly, cross-sectional is getting data on an individual at one time point and seeing how people of different age ranges compare on that data. Longitudinal is following the same people over time. So the challenging thing about longitudinal is it takes time for people to get older. And it's something that, we've been doing painstakingly for the last 6 years, is waiting for people to get older to see what happens. But in the meantime, we can look at cross-sectional age relationships and how those age relationships might be different between autistic and neurotypical individuals. So in the early days, we looked into this fantastic resource called the Autism Brain Imaging Data Exchange that has open access data of MRIs from about 1,000 autistic individuals and neurotypical individuals. When we looked at the adult participants in this data set, one of the kind of shocking things that provided further credence to how important this work was is that less than 3% of the participants were over the age of 39. So this really shows, when you collect the brain imaging research that was being done all over the world, how few people were including older subjects. So we ended up adding 47 of our own participants over the age of 40 to make strong conclusions about age relationships in this brain data. And the measure we were looking at here is looking at the cortical surface of the brain and how thick that tissue is, which generally at older ages, at least, we think of as thick is better. Thick is more neurons, more synaptic connections. And the program we use for this is called FreeSurfer. So we were looking for areas in the brain where the thickness changes with age, but differently between autistic individuals and neurotypical individuals. So when we ask this question of the data, we see some hotspots here on the brain image to the left. And then we can plot the data in the hotspots, which is the graph on the right. So on the graph on the right, we see that we have age on the x-axis, that age increases as we move to the right. And cortical thickness is on the y-axis, where cortical thickness decreases as we move down. And the ASD group is in red, and we see from young adulthood, 18 up to about 65 in this sample, that indeed the older individuals are showing thinner cortex, which is what we would think of as more indicative of perhaps neuronal loss and synaptic or connection loss. Whereas in this relatively middle age range, the neurotypical group is pretty stable. The neurotypical adults who are in their 50s have about the same similar thickness to their cortex as the younger neurotypical adults. So this is cross-sectional research. It's not the same thing as following people over time, but it is a window into aging that perhaps the cortex might be thinning faster in autistic adults. And these hotspots that we see are really concentrated around the temporal lobe. And the temporal lobe is really important because it does our memory functions. That hippocampus structure is kind of folded up inside the temporal lobe, and the outer parts help the hippocampus. And then it also does some really important language functions. So this was something that we wanted to follow up on and pay close attention to. And we were able to, with this data, get funding from the National Institute of Mental Health to continue and expand our aging study. So here we were able to start including women into our study. And we also recruited our own young adults cohort to do these cross-sectional comparisons. So the table on the left is a little bit busy, but shows you kind of how we're doing with how many participants we have, what are their age ranges. Again, everyone is without ID in this sample by nature of the brain imaging tasks that we do, but it is a very important future direction of ours to do what we can with brain imaging with people with comorbid ID. And then I think the graph to the right is a little bit easier to digest. So this is every person that's in our study plotted by their age. And then as you move to the right, you see that people that are over 40 years of age, we try to get them to come back every 2 years and collect all this data on them again to see how things change as they grow older. So some people we've followed 4 times now over 6 years. And one of the first things that we wanted to do once we collected enough longitudinal data was to attempt to shed light on this discrepancy in the literature, where there's some papers that show, at least cross-sectionally, autism might be associated with some protection to cognitive aging. And other papers show the opposite, kind of more similar to our brain data, where older adults with autism are showing, reduced cognitive function compared to their younger counterparts. And these pose really interesting questions about, how different of an experience is it to be 70 years old now and lived a life as an autistic individual versus being 20. And that's what we call cohort differences. And then if we do see this profile of sort of protective aging, where our oldest autistic adults are doing quite well cognitively compared to younger, what kind of resiliency is at play there, and what kind of compensatory strategies are making that possible? So this is a graph that shows kind of that protective aging profile. You can see the lines are the exact opposite of that cortical thickness brain data I showed before. So this measure is short-term verbal memory. And you can see as age increases, our neurotypical group that's black and gray, their scores are going down, which is what we would expect for memory. Older individuals are not performing as well on memory as younger individuals. However, you don't see that pattern in the ASD group. The oldest individuals who are in their 70s, late 60s, 70s, who are autistic are doing just as well as the younger autistic individuals who are in their 20s. So this is kind of the pattern that Hilda Gertz has published a few times that shows kind of this protective aging pattern. So when we followed these same people in this graph who are over the age of 40, again, we were able to compare within their own performance. Are they getting better at memory? Are they staying the same? Or are they getting worse? And that's what this data shows here. So we were able to categorize people as either improving, being stable, or declining. And unfortunately, when we look within individuals, we do not see any evidence for protection against cognitive aging associated with autism. We kind of see some evidence of the opposite. So when you look in that decline category to the left on the x-axis, the individuals with autism are in red, and we see significantly more middle-aged and older autistic individuals declined in their memory performance from one time point to another time point about 2 or 3 years later. And that wasn't the case for most neurotypical individuals, but it's important to point out that most autistic individuals as well remain stable. It was about a third of the autistic individuals that showed this significant decline in memory performance. And we've also looked at longitudinal change in hippocampal volume, where we see a similar picture. So this is that brain memory structure that the ASD group on average has a declining trajectory over 2 to 3 years in hippocampal volume. It's getting smaller. And the neurotypical group in this age range is staying relatively stable. And when we include data of people that we've followed 3 times, up to 4 to 5 years, we see a pretty striking difference in long-term memory now. This is visual long-term memory, where the ASD group in red on average is declining in this long-term memory measure, and the neurotypical group in gray and black on average is staying relatively stable. So a summary for this kind of descriptive cognitive and brain aging research is that indeed there is this cross-sectional effect where older autistic adults are doing just as well, if not better, than younger autistic adults on cognitive measures. But that there's no evidence, at least in our data, for this when we follow the same people over time. And granted, not everyone's declining, only about a third of our participants. And this still is a relatively small sample for a relatively short follow-up duration. So we need to continue following these people and more people as they grow older to get a clear picture of what's happening. When it comes to the brain, it's not as confusing of a picture. For the most part, both cross-sectional and our longitudinal findings are showing that there are vulnerabilities with how the brain might be changing faster in autistic adults as they grow older. And as I mentioned before, these findings bring up really important considerations for what are the successful compensatory strategies that older autistic adults have learned and are employing as far as cognitive abilities goes in comparison to younger autistic adults. How can we figure that out and capitalize that and infuse that in perhaps some cognitive compensation intervention strategies? And then as an aging researcher, I'm extremely curious about what it is about being in a different cohort, an older autistic person, and the experience that they have lived that leads to this different cognitive profile compared to younger autistic adults. So now we'll switch gears and start to talk about some precision care considerations. So the first topic I want to touch on is about trying to identify who is most vulnerable to this accelerated cognitive and brain aging profile. Who is most vulnerable to decline in their memory and who's more likely to stay stable? And how can we use that information to provide treatment plans and appropriate interventions? So I'll talk about a baseline brain measurement and a couple of genetic factors. So when we think about memory, we think about the hippocampus in the brain. And I have presented this a few times. The hippocampal volume, the total size of this memory brain structure, could give us insight into who's most vulnerable to memory decline. Perhaps people with smaller hippocampi at baseline are more likely to decline in memory. But then I'm going to also present data from this newer MRI metric that's called free water. And we get this from diffusion imaging. So this is a measure of how far do water molecules move in the brain before they bump into something. And when we think of a brain that's packed with neurons and packed with connections, water doesn't have very far to move. It's not very free. But when a brain has lost neurons, lost connections, there's more free water. And this is more indicative of like micro degeneration that could happen earlier than the type of degeneration we would see in total size of the hippocampus. So indeed, we find it's this free water metric that actually correlates with future memory change. So in the x-axis here to the right are people with the most free water, who probably have the most degeneration, and they are showing the most memory decline. So we can actually look at their free water at baseline and with decent, significant, at least accuracy, predict who's most likely to decline in their memory in the next 2 to 5 years. For genetic factors, as an aging researcher who's worked in Alzheimer's, the main genetic factor we think of in aging is a gene called ApoE. It's the biggest risk gene for Alzheimer's disease, but also a host of other health outcomes. So this gene comes in a few different flavors, and there's one flavor that is associated with a risk of Alzheimer's, and that flavor is the 4th allele. So in our own cohort, we see that autistic adults are more likely to carry this Alzheimer's risk gene than the neurotypical adults, about twice as likely. And another group has published this as well. And then looking how this influences brain measures that we have on our cohort, we look at that hippocampal volume size, the size of the brain memory structure. And it's only this group with the red bar that actually has smaller hippocampi. And so this is the group, the autistic group, that has this risk gene for Alzheimer's. So perhaps there's kind of an interaction here for being autistic, but also carrying Alzheimer's risk genes that's going to be really important to predicting who's most likely to go on and develop dementia or accelerated memory decline. And then finally, we've done a little bit of work into genomic measures that can be investigated across the lifespan and could shed light on future prognosis for memory decline. So telomeres, what is a telomere? It's basically the ends of the chromosomes. It's non-coding repetitive sequences that sort of protect the coding regions of our chromosome. And these protective regions at the end get smaller as we get older. And one mechanism is cellular division. But they're also associated with stress quite strongly. And they're associated with age-related cognitive decline. Shorter telomeres in older age is related to a worse cognitive profile, higher risk for dementia. So we and others have shown that even in childhood, autistic children have smaller telomeres than their typical counterparts. So there's a lot more work to be done here, but it certainly begs the question of, if your telomeres are smaller in childhood, does that put you at risk for dangerously small telomeres that are going to be related to cognitive function as an older adult? And could we measure telomere length even in kids and give some prognosis of cognitive development? So to summarize those few points, we have this exciting data about hippocampal free water that might be an MRI brain metric that could help us predict who's most vulnerable to memory decline. We have some insights into a very well-known Alzheimer's risk gene and how this might be interacting with autism to adversely affect memory brain structures. And then finally, we're paying attention to telomere length and how that could set up an individual to be at greater risk for accelerated cognitive decline later in life. So the last little bit I'll touch on is still in the realm of precision care, but this is thinking about interventions and who's most likely to benefit from an intervention, so that we're not wasting time, we're not wasting resources, we're precisely matching someone with an intervention that they're most likely to respond to. And this is going to be important for all interventions, but we focus on behavioral interventions. And there's a lot of factors that go into this. And the two main factors that we focus on to date are sex and gender and age. So first, to talk about what it is that we target in our interventions, and it's quality of life, which is probably the most consistent finding or one of the most consistent findings in the adult ASD literature, is reduced quality of life in various domains. And we've published on this and the influence of sex and age, where we show that both autistic men and women have reduced mental health quality of life, whereas only autistic women have reduced physical health quality of life. And then cross-sectionally with age, we actually see this really encouraging finding that, at least for autistic women, mental health is better in older age ranges than younger age ranges. And the approach we use to try to improve quality of life is a program called Mindfulness-Based Stress Reduction. So this was brought into the research community out of UMass by way of John Kabat-Zinn. And it's been widely used in many, many clinical and non-clinical groups, with meta-analyses of thousands of individuals that show that it's quite beneficial compared to active control groups with small to moderate effect sizes. And the theme of this program is to try to teach individuals adaptive coping strategies that allows one to redirect their focus to what's happening here and now in the present moment, and to do that without judging it. And the structure of the class is to meet weekly for 8 weeks for 2 hours at a time, with practicing the meditations and mindfulness at home every day. So we first looked into this as being a potential benefit for autistic adults. There was one group in the Netherlands who was already doing an adaptive MBSR protocol. And their approach was that the teacher was both an MBSR credentialed instructor and also an ASD clinician. And they found quite good effects in several papers for reducing depression and anxiety symptoms. But some of the important limitations that we saw is that finding someone who's both an MBSR instructor and an autism clinician is kind of impractical as being an option that we could make widely available to the community. And they had not yet compared the effectiveness of MBSR to an active control group. So a control group that received social support, for example, which we know could be extremely important for autistic adults. So our approach was to do an MBSR intervention, but do two instructors, one who's a credentialed MBSR facilitator and one who is an ASD clinician, but with some MBSR training so they could kind of work together for maximum benefit. And then we had an active control group that had social support and also received some relaxation education. And they met for the same amount of time and they were told to think about this and practice this every day. So I'll show a little bit of data on about 30 participants in each group. A little over a third were women, and we have a broad age range and broad IQ range, albeit everyone was without intellectual disability. So the good news is that both interventions improve mental health quality of life, but we did see some MBSR specific improvements in disability associated quality of life, where the challenges posed by one's disability decreased in the participants who learned mindfulness meditation. And then getting at the precision medicine piece, the big factor that jumped out at us was a gender difference. So in both groups, it was actually only the autistic women who improved. So first looking at physical health, our men are the lines up top and fairly stable, whereas our women in both groups improved on physical health quality of life. And granted, men weren't struggling so much with this measure, so it could have been a ceiling effect. But this is very salient when looking at mental health, where, again, our men in both groups are staying stable pre to post, whereas our women in the blue and the red are improving in mental health. So this really suggests this might be most beneficial to autistic women. So I think I'm pretty much hit the summary that, we have these encouraging findings about quality of life with these behavioral interventions, really expands options for autistic adults when we find that just a social support group that talks about relaxation can be just as beneficial as mindfulness, and that women might respond better to these group-based behavioral interventions. We do need to do more research into how other gender identities respond specifically, like non-binary individuals, which we didn't have enough participants to be able to look at a group of other gender identities. And IQ and age didn't seem to be an important factor in response to these intervention groups. So to bring everything together for some preliminary conclusions, because this is still very much ongoing work, on the cognitive and brain aging side, we do see some vulnerabilities for accelerated memory decline and accelerated changes of memory brain structures. But it's important to remember this is not deterministic. This is not happening to every autistic adult. It's happening to some autistic adults. And we see some encouraging findings of resiliency with age, that autistic women are doing quite well with their mental health, and trying to figure out what it is about older age for autistic individuals that they've come up with compensatory strategies to do better in some cases than younger autistic individuals. For precision care, we have this really interesting MRI metric of the hippocampus that might give us insights into who's most vulnerable to decline. And we also have some genetic factors associated with Alzheimer's and DNA protection. And then on the behavioral side, this really compelling evidence that this group-based type of intervention that includes mindfulness or relaxation might be a really good option for autistic women. And of course, I'd like to thank my incredible team at ASU and our incredible partners around the Phoenix area, our participants who are so dedicated to this work and this study, and our many funding sources.